Okay, let's recap where we stand. We can predict, in principle, proteins fold from scratch in solvent. Solvent is, for protein, the native environment is water. I said that its density in a cell is like a solid state, but the protein is still surrounded by water. This detail of the way it folds into a three-dimensional structure is relevant for function. We can find out about relation of proteins through comparisons of the sequences, because this is what evolves. The sequence in evolution, the sequence changes. You pick up the evolutionary signal, you do it in a pairwise alignment, sequence to sequence alignment, by simple brute force, dynamic programming. That's one way of doing it, and speeding it up is hashing. Again, a typical question could be, why do we have to speed it up? Proteins are 400 residues long, so doing a 400 against 400 comparison matrix is easy. Why do we need to speed it up?
Because there are 100,000 nodes.
So the point is, we are not really interested in one pair. We want to know anything about a particular protein that has been discovered today. Say there's a sequencing, so somebody digs into the soil and picks out what they find, and they sequence that, and they find some protein in there, and they want to find out whether that protein is related to whatever is known. Then they will run against 60, or at this point over 80 million sequences. And that's the process that is slow. What is very slow is to do an all against all, so 85 million against 85 million. That's very slow. And even with a hashing algorithm, this is beyond most of the computers. Currently, this is really difficult to do. So all the groups that have to do this type of job, including ours or some subfraction, we live through hacks, shortcuts. A more refined way to compare sequences is by building up these pairwise alignments. For a family, you build up a family, the family defines a profile, and then you align a single sequence against a profile. In terms of CPU, is sequence as fast as sequence profile? Anybody in the... Any idea? Okay, do you remember how it's done? How do you compile sequences against profiles? Okay, let's just keep it there. You may want to look that sort of thing up. Profile comparison is a step deeper. So you build up families on the search sequence and the queries on the database, and you do a profile against a profile. And that allows us to go to identify relations that are very distant in terms of sequence. What does it mean, distant in terms of sequence? Is that clear, yeah? That is correct. So the pairwise alignment is very, very low, and very low could be, to just throw a number at you, 10% sequence identity. So 10% of the residues are identical in 400, say. Now, and still structure and even function may be similar. How can that be? Again, I told you that many proteins that have 10% sequence identity will not be similar. How come some are similar? How could you imagine this? How could you explain that?
The environment where it is located and the evolution.
Essentially, he says the environment where it's located and the evolution. This is very related. So evolution, essentially, so you have random changes of sequence. And some are selected for by the environment, by the function, by essentially what this thing has to do. And some are not selected, so it's selected against, meaning they will not survive, or the organism that carries it has a lower chance of survival to pass on the genetic material. So, or the protein will just not be expressed. And so there's a variety of mechanisms by which this can happen, but ultimately the result is what works, changes that either are neutral or gain something, they stay, and things that kill it, or kill in the sense that make this function no longer work, will not survive, okay? And this means you have a selection for maintaining some pairs with very distant sequence identity and still maintain function, because there's a pressure to maintain function, there's a pressure to maintain structure. Whenever you compare two proteins with 10% sequence identity, they may not be related, but when they are related, evolutionarily, when they have a common ancestor, or when they come from two organisms that have a common ancestor, then there is a certain probability that in fact they are functionally related, and part of this evolutionary process of selection to maintain function. Why is sequence changing? Why protein sequence is changing? I'd like to have somebody from the back more. Sorry. Yeah? Mutation. So yes, but why are we mutating? So when I'm sort of running around here, are my proteins mutating?
I could imagine that there are like lots of reasons, like
radiation,
replication errors.
Replication errors. So replication, you have more?
Viruses, perhaps.
Oh, viruses, that's, yes, but that is a very, very infrequent event. So radiation is way, way more likely than a virus changing my genome or my protein. No? Viruses typically attack cells and sort of set their own DNA into the cells and make the cells become their slave to express their DNA and actually produce viruses. But they typically don't lead to mutants, although that does happen. And CRISPR-Cas, so the modern way of genetic engineering and hopefully curing diseases, is related to that sort of idea. But okay, let's go for the first ones. Radiation. So there is a chance that radiation in fact changes my DNA and therefore the underlying protein, and that does happen. It's a relatively frequent event. There is aging, which leads to mistakes in the way it's copied, and there are regular copy mistakes. Now regular copy mistakes, there are many of those. However, since there are so many, there is a mechanism in place to somehow correct for them. Radiation less so, and aging also less so. So there's a variety of errors building up. And some of these errors are in fact, or changes, let's call them variants, pass on in evolution or in your family. Do you have any idea, and this is off topic, do you have any idea how much we differ? Again, everybody of us has about 20,000 proteins. And the protein has about 400 amino acids or residues. Any idea how many differ between you and the one sitting next to you, provided this is not family? So any unrelated two in this room here? Unrelated again, it's family related.
I think I heard it's something like 98 to 99%.
So 98% or 99% is identical, you say? Yeah. It's actually more. So that would be shockingly low, because somehow in the news, in typical TV shows, you may see things like mouse and human is related to 98%. It's actually not true, 95. It's a very misleading way of looking at it. But the way we think about it is that in every pair of us, in one protein, there's one change of an amino acid. Okay? There are many other changes that are not visible on the level of the protein sequence, but one in every single protein. And this is actually much more than we had assumed. And these changes seem to have much more impact than we had assumed. We just published a paper on that. Okay. We cannot predict 3D structure from sequence. The protein knows how to fold. In the computer, we cannot repeat that because of the complexity of the folding. So yes, the protein adopts a unique three-dimensional structure on its own, but we in the computer cannot and do not manage well enough the energy functions and the complexity of the problem in order to repeat that in a computer. That means we cannot fold a protein in a computer. We can fold small peptides. We can sort of fold some periods of the time that a protein folds, but predicting through the process, through simulating the process of molecular dynamics, for instance, the thing still does not quite work, certainly not for, of these 20,000 proteins, 19,990 or something. Maybe for some it does work. It does work for some. The only way we can use the computer to predict 3D structure is comparative modeling, reliably. Comparative modeling simply uses the feature that two proteins with similar sequences have similar structure. So I align against 100,000 for which, or 120,000 for which I know the three-dimensional structure. A protein of unknown structure, I align against the ones that I have the experimental structure for. 120,000 of those are in the database. And if I find one that is similar, I say that has the same structure as the one that I'm querying with. And that is comparative modeling. Comparative modeling at this point puts about models for about 40 million or more proteins into the database. But for most of all the human proteins and for most of the residues in the human proteins, I still do not know, cannot reach by comparative modeling. So for those I need something else. And for an extreme way to do something else is to essentially predict 1D structure strings, and one example of 1D structure is secondary structure. I talked about two generations of methods. The first generation that simply looks in some way at single amino acid propensities. Single amino acid propensities simply ask what's the probability that a particular amino acid is in one of the three secondary structure states, helix, strand, or other. I build up a probability matrix, and there is a variety of ways in which I can do that. I can step up the intelligence of doing this in the second generation of methods, where I look at windows or pairs of residues or k-mers. We discussed that k-mers would only work until possibly a k of 3. Maybe 4, but most likely not 4 at this point because the database is too small. And we discussed in the last lecture on last Tuesday, no, I'm a little bit off, last Thursday, we discussed that there are some problems with these approaches. One problem is they don't reach 100% accuracy. One problem is beta strands are not predicted very poorly. And another problem is that segments are predicted too short. Then I talked about a machine learning approach. This particular machine learning approach is a neural network that uses, on the left-hand side, the amino acid sequence as input. The yellow ones are 13 times 21 input units. 21 because you have 20 amino acids plus a spacer. The spacer, bless you, the spacer is needed because you shift this window. Say my hands is only 5, but up there is 13 residues. Say the middle finger here is the P at the center of this window here. And when we shift this through the protein, at some point the P should be the first residue. So there's something that is not an amino acid before the P, that's the spacer, hence 21 units instead of 20. So 13 times 21 input units. What I show here is arbitrarily 6 hidden units in blue. So if you have enough information, then the hidden layer, so the number of hidden units is typically larger. Initially, when this was first done 20 some years ago, in fact there were 15 input units, and I just drew 6 here because it's simpler to draw. The output really is 3 states: helix, strand, other. And I showed you that this device alone doesn't solve all the problems, but by sort of interfering with the training dynamics, we could predict beta strands as accurately as alpha helices. Somehow at the cost of the overall accuracy, but this is one severe problem that the machine learning, by understanding the way machine learning works, could address. A second issue that we could address is by sort of combining 2 of these networks. So this one here is what I just described. The secondary structure as output, 3 states, and the output of this first level there goes as input to a second level. The second level sees correlations between adjacent residues. Would, for instance, see that in some cases, on the first level, you would predict, so I have residue I plus 1 and I plus 3, H, L, H, L, H. And in another case, I would possibly see H, H, L, L, H. And maybe it would learn that in this one here, you have to really convert all the H's into L's. And in this one, it may be the best thing to convert the L's into H's whenever it sees things like that. You could now imagine to increase the complexity of this device by, rather than using, as shown here for 3 consecutive residues, 3 times 4 input units, or so 12 input units, to put some additional information in there. What could you use as an additional information in the second level?
Just to refresh that to understand correctly.
So question is hold it, here's a question, another question, yeah?
So you train the leftmost part separately, right?
So the beauty of this, let me repeat the question and answer the question at the same time for the people in the back of the room. So the question is, are these two devices, so this is essentially one device here and that's the other device, are these trained independently? So yes, the beauty of this device here, the beauty of sort of stacking machine learning approaches in this way, as opposed to in deep learning, the upside is that you sort of have independent devices and you control how well they perform independently. That's the beauty. The downside is that in the deep learning approach, you have sort of one entire algorithm threading through the whole thing. And that you lose, because you decouple these two devices. Yeah?
Follow-up question. So this 3 here, but potentially 21 copies
Yes.
are just copies of the same thing.
Yes, so the 3 that I show here, and again in the first implementation those were 21, or whatever, 3, I show 3, they are a copy of the same thing, right? Copies of the same, so you train this device here on the first level, so let's go a step back here. This essentially is one neural network, and you train this device for the entire data set you have. The entire data set, say you had 10,000 proteins, 10,000 times 400 residues, so you had 4 million data points to train on. 4 million for which you know the HEL state, and you train by presenting the samples and by changing the connections, shown here in green, such that the output that the system gets resembles what you know it is. So for this particular P here, you know it happens to be in the state E, extended, so strand. And you want the system to get that out. The thing that you can change in the network, the only free parameter that you have, or the major free parameter that you have, is essentially the connections, right? And you change the connections such that for the known examples, it creates what you have observed. And you hope that by doing this, the system has learned implicit rules, has learned generality of the problem, and in fact will work for proteins of the future that you haven't seen before, that you don't have in your data set. Okay? Now, the way this is trained is you present one sample at a time. Every single time you do in an energy landscape where you plot the connections versus the error, in this landscape you go downhill. So you change the connection such that with every change you have a little bit less error. Okay? And sometimes you allow uphill moves. Now, every single time you present one example, the way you choose the examples is you take your 4 million into a database and you randomly pick points. So you pick it from here, just like I would pick randomly people in the room. Not neighbors. Okay, you're not threading an entire protein through and say protein residue I plus 1, I plus 2, I plus 3. You'd pick it from protein 1. So I pick, that's the first one, that's the second one, that's the third one, and so forth. I have to randomize that, in fact, to avoid correlations. And then, yes, I create one device. And this device here I use to predict the secondary structure of an entire protein. I simply run this through, right? I shift the window of, shown here, 13 residues through the protein. If the protein has 100 residues, I simply run the same thing 100 times. Okay? And this is essentially what this means here. This is 3 times the input here, 3 times the same thing. It's just, I said I run it 100 times. In this image here, I run it, the leftmost, I run it 3 times. Okay, so now the question, is that question answered? So now the question that I asked needs to be answered still. So now I have the second level here. And again, just visually, if I want to do 21, what I have is here is 3 sort of copies that I have 21. My question is, how could I put the second level here? At this point, all it gets as input is the sequence to make decisions such as this one, these alternatives. Now, could I put more information in? Maybe whether or not I move the else or here the else has to also do something with something else. What else could I put in? No idea?
Can you please clarify what do you mean by something else?
Well, at this point, the second level is a neural network trained completely independently, and all it is trained on is the prediction of the first neural network, right? And it's an independent device. So since I use a certain set of input information, I could use another piece of input for information. What could I use? Okay, I could, yeah?
Have the average length of links?
You could look. That actually is a great idea. That actually never occurred to me. So let me repeat that. So this is a totally great idea. I never did that. I don't know why. So the idea here was, why not use, not in this particular case here, or in this particular case where we have 5, you see something about a local window of 5. But you could also put in in the local run in which this sits here, in this local run, the longest h is 2 and the longest l is 2. And the average is 10 or 11 for h and 5 for l. So you could put both into the input, or you could put the difference into the input, or you could put the ratio between the two into the input, and that would be a great idea. And it would be very simple because it's relatively little additional input units. So you don't add to the complexity of this neural network here, and you put clearly relevant information in. And I never thought about that. This is a great idea. Yes?
So this is, do I get it right that this is technically redundant information, but it helps network to learn?
So in this particular case, so far it's not redundant. Okay, the question is that redundant information? So far it's not. Because so far, all that the second network, I'm actually asking a question, and the answer to that question could be that it would become redundant. But at this very point, the second level sees a correlation that the first one can never see. The first one will only see the secondary structure for one individual residue here. Let's go to this initial network here. In the brackets, I show the secondary structure state of that particular amino acid. But the system doesn't see it. All that the system sees is the P here is supposed to be in a state, and if that is corrected, or if in the default configuration of the connection H is predicted, then the system will see that this is wrong. But it does not explicitly see anything about the neighbor secondary structure. Okay? In that sense, the first level and the second level have completely independent information. Not only are they trained, they are also not correlated. Or they are only correlated, if in the sense, if you solve the secondary structure prediction problem, then they would be correlated implicitly, but not otherwise.
This is clear. I'm asking if you're adding, for example, average length of the sequence or something like that.
Sorry?
If you're adding something, the question that you asked, can we add something on
Yes.
top? So at this point, when you add something on top of it, does it become...
Okay, so fair enough. Cannot somebody answer the question first before you ask the question, what if I... So he's now asking the question, what if I edit in the second level sequence? It's not quite what he asked, but implicitly that's what he asked. Then, would I not introduce sort of a correlated piece of information into the second level? That is correct. And that, in fact, my question, that's what I wanted as an answer. We could also put into the second level the sequence again, or some fragment of the sequence. So your idea of the average length of a segment is quite a different one, but we could put some redundant information. Since the second level sees something else, the redundant information may still be helpful. And I don't really have to give the same type of information as in the same level. So maybe not the entire sequence, maybe only some fraction of it. Okay. Again, it solves with this machine learning device, if we handle it correctly, solves two issues. It predicts beta strands better, and the length of a segment is looking very realistic. But we are still only getting something like 60-something% accuracy. How can we improve? And that brings us, so this was the recap, that was the introduction to secondary structure prediction. The third generation of methods, and today will be about the third generations, is sort of improving, answers that question. Before I get into that, are there any other questions? Okay. So then, yes?
So what's the accuracy we can get with cooperative modeling as like...
Oh, that's a great idea. That's a great question. I will get back to that. So the question here is, what is the accuracy that I can reach by comparative modeling as compared to secondary structure prediction? Historically, and I hope that I give the answer, otherwise remind me at the end. I hope that there's a slide giving the answer explicitly. So historically we assumed that comparative modeling would be much better than this. If we can get a 3D structure, then assigning the secondary structure from it will be better than just secondary structure prediction. That's what we had always assumed. But the reason for having secondary structure prediction methods is because when we cannot do comparative modeling, when we look at residues where there's no comparative model available, then this is the only thing, or that's one thing that we can do. Okay? But the assumption is that comparative modeling is substantially better. The second question?
Going back to the first level of input, the network gets, is there already a probability assigned to each amino acid? Like phenylalanine is probably
So that's this sort of is an interesting idea. And in this particular case, I'm not entirely sure how to name it. So let me repeat the question. So again, the way this works here is the black means that at that particular position there is a p. 1 is 1, or 100, or whatever your scale is, and everything else is 0. Now, since we do know what the task is to predict secondary structure, we could actually, rather than putting in a 1 or 0, which is a count of that amino acid, we could somehow multiply that with the expected accuracy of this P to be in a helix, right? Is that what you mean?
Yeah.
Can you see what the problem of that could be?
This is kind of inductive bias.
This is kind of...
Inductive bias. You're like influencing learning by whatever you're biasing it towards something that...
So the problem could be that it could be. So machine learning is like a cookbook. Typically, you try it out and then you see. And in this particular case, the one that I know who tried it did not believe it would work. So that means that person was also biased. And maybe what that person found was sort of what that person expected. And that happens very often in science. But I found this not to work. And I rationalized that it didn't work because the simple probability models are much less accurate than the machine learning device. And by putting this initially in, the network could never get out of the trap, so to speak. It kept. In fact, this did not even learn the beta strands right, at least in my hands. Now, this being said, what you could try, and when I did that, I just did not have enough samples, what you could try is to use both. Meaning you leave the network the space to benefit from it and to ignore it. If you have enough samples, then typically this is a very good idea. Anything that you believe can help, if you have enough samples, you put in. When I did this first, I just did not have enough examples for that. There was another question. The question moved. Yes, the question has moved. Yes?
Do you normalize the data? Because I feel like the base order would be much more common than others.
How would you normalize? So the question is, do you normalize the data? How would you normalize the data?
That's the question.
So I'm not entirely clear what you mean by normalize. You have to say something more. So one question is, so let's state the first statement. And let me, before you go on with your question, let me bring it to the room. If the circle represented the 120,000 structures in the PDB, protein database, do I take them all? You all should be able to stand up and shout, no, absolutely not, there's too much bias in biology and there are many reasons for that. And that type of thing is true in biology, but it's true anywhere else. You cannot take the data at face value. You have to sort of massage and filter the data. You have to begin to understand your data. And that means typically that you have to see whether there are implicit overlaps in the data. So we cannot take everything. And when I said we have about 10,000, then that's a sort of redundancy reduced subset of that. 10,000 here are things that we know are accurate in terms of, more accurate in terms of experimental structure. They are such that no pair in this set of 10,000 can be used to do comparative modeling. So in these 10,000, there is no pair in the 10,000. When I do an all against all, 10,000 against 10,000, there is no pair that is within the homology distance. So that's a redundancy reduction. And in some sense, that is what you may call a normalization. Now beyond that, what do you mean?
Aren't the single amino acids less frequent than others?
So you want to add into the system some knowledge about how frequent that amino acid is in the database or in that protein?
Yes.
Both? So the database doesn't work at all. What is actually interesting is, and you have to put that as an implicit thing, and again I hope, I'm not entirely sure how my lecture continues today, I may have skipped this. That may be the second thing I have to remind me when I stop for questioning. I'd like to, if my slides don't get there, I'd like to get back to these 2 points here. And so putting the difference between what is my protein, the composition of my protein, so is my protein more proline rich than the average protein? So that's relevant information. That helped. But I'd like to say something else on that issue and on the comparative modeling issue, yes?
You mentioned something surprising to me, that in every protein, for any pair of individuals, there is a difference, right? In every protein.
So, I'm sorry. Now we get beyond the second. I foolishly got into a different topic. And that different topic is essentially how is the variance between us affecting our differences in the way we look, in the way we react to particular drugs. So why is it that I need to possibly have a different drug against headache than some of you? And that has to do with the variance between us. It has to do with other things. It has to do with the microbiome that I'm carrying around. But the variance between us are relevant. And I said that about one amino acid is different between any pair in the room in every single protein. Now, this is not meant to mean that every single protein for everybody has a difference. It meant to mean we have on average 20,000 between any pair in the room. Since we have 20,000 proteins, that the number that I sort of visualize is 1 per protein. Does not really mean it has to be 1 in each protein, okay? In fact, they cluster, and they cluster differently. But so far we haven't seen, so in the clustering we also learned something about proteins. But this is much beyond the lecture. Is there anything else you wanted?
Just ask a simple question. For example, we have a hemoglobin sequence in PD.
Yeah.
And there is only one.
Yeah, no,
So,
there are many, but...
I mean, if my hemoglobin, your hemoglobin, and I don't know, hemoglobin of everybody in this room are different by 1 amino acid, can this sequence in the PDB be considered the ground truth? Or maybe this is...
And that now is, I really should not have talked about sequence variation, but I do want to talk about it because this is what our lab does all the time and it's totally cool. So, when the first... Could we briefly cut the camera? Maybe it's next semester. Let's call that a step for next semester. So the question really is, we need to somehow get more information. So we cannot get to 100% because somehow in this window, sliding window approach, the information that I get is not enough. Yes, I reach a certain level of accuracy, I can solve all kinds of problems with machine learning, but I need to somehow get more information into my system. How can I do that? Any ideas? What could be the third generation? So we have a first generation where we look at individual residues. We have a second generation where we look at consecutive or pairs or couplings or local density. What could be the third generation? Any idea what you could put into the system? How can you put more information? So ultimately, again, the problem is my window of, say, 21 residues is not capturing enough information.
So each beta strands consist of like some subchains or which are distant from each other. Maybe you encode the information of this relative positions of the sub.
So what he says is, okay, beta strands, I talk about the E state in my slides always. There are two beta strands that come together and they form a sheet. They touch. So what about putting the information about the bonding sheet into the story? And that is a great idea. Now here's a problem in that. I try to simplify my problem, right? I try to say, okay, there's 3D, 2D, 1D, and let's first begin with 1D prediction. If I now use bonding, then I'm back to this 2D problem. And the 2D problem I told you, if I can predict contacts or distances between amino acid residues in the protein, then I can essentially predict the 3D structure. Meaning that if I really solve the 2D problem, I actually solve the 3D problem. And I'm trying to do something on a lower level of complexity. So this is not quite simple. Nevertheless, people have tried that, in particular for beta strands, because the feeling was the bonding is so strong. But unfortunately, the bonding predictions was not good enough to help in this. So this has been tried. That did not work. Does not mean that it doesn't work in principle. It's just that the solution or the kind of constraints we got from the beta strands that we know, so from first predicting which strands would bind, was not good enough to help in the other one.
So you want something along 1D?
I want to predict 1D structure better.
No, I mean your additional piece of information. Should it be also in 1D, like relative position?
So the question is the information that I want to put into the system 1D? Well, the information that I want to put into the system is such that it brings me more about secondary structure than I have. The information is such that I need to have it on the level of sequence. So in that sense, it's 1D. But you could possibly massage this 1D in a sense in which you get more information. Sorry I haven't seen you.
You could maybe try to add related sequences into the prediction.
So this is 99 point at 100, because it gets a washing machine or something like that. Congratulations. This is a first. Wow. So the idea is essentially this one here. When I showed you this slide first, number of residues aligned, percentage sequence identity, I said everything in the blue area is within the comparative modeling distance, or means that if in 100 residues you change 33, keep 33 constant, so you change 67, then the two things essentially have the same 3D structure if they evolve the same. Now, if I went for the same protein into the lab and I picked randomly 5 residues, it is very likely that I change structure, okay? So how come? There are 67 exchanged here, and I cannot even do 5. Yeah, is that a question or an answer? So you say a couple of things at the same time, and it's very true. So let me repeat what he said. So there are rules, or there are reasons why some can change and some cannot change. They're implicit. There's some information implicit in the way of change. And that is absolutely true. The first statement is it can happen because evolution, again, selects the ones, so it selects the least likely thing. The least likely thing is a variant that does not have an effect. So the 5, if I changed randomly 5, the probability of change is pretty high. In evolution, what we see is the unlikely event of neutral variance. And the unlikely variant survives. All the others die out, okay? What we see is the result of a long selection process against the things that don't work. Hence, in the lab, I cannot do what evolution can do. And indeed, once I have this pattern, it contains information. So once I have a family, at some points here in purple, the protein is more conserved, the family is more conserved, and if I look at these purple residues on top of a 3D structure, these are two different sides here of the same molecule, you see that the first one here and the second one are close in space. Now, when anything happens within, say here on this window of 5, so this rectangle here cuts out 5 consecutive residues. And anything that happens within those 5 residues has an influence on what happens here and is influenced by what is here. Okay? So what you will observe in these 5 depends on what is here. That means that if you sort of take this profile, if you abstract what you said, if you took these rules, these implicit rules for 5 consecutive out of the profile, you have somehow a view of what happens somewhere else in the molecule. Because what you observe here depends on what is here, which is far away in terms of sequence space. Far away in terms of sequence space, what I mean is, if you cut out a window, a sliding window of 13 or 21 or whatever, this is further away, okay? That's the point here. And this implies that you somehow convert a problem that is sort of the one that is on the level of chaos to a problem that you can actually solve by averaging over different species. And that's exactly the idea. So the way you change that in the system that I showed you is very simple. Once you have this idea, how to put it in is totally trivial. You simply build up the family, and I showed you that before, so the sequence runs down here now, and what we have here, 4 related sequences from a family. And at the central proline here, it happens to be true that everything in this family here has a P. At some other positions here, you see other residues in that family. And now, instead of each one of these blurbs, and sorry, they have changed the color, that used to be the yellow one here, and that used to be the blue, and that used to be the red. Let's not dwell on that. So before, they were 20 zeros and 1 1, every one of these 13 blobs here. And now each one of those essentially has a value between 0 and 1, so a probability or a frequency of observation. So how often in that family do I see the glycine? And so forth. And, so here I have an example. So the entire information is carried here by the D, while here it's split between the A and the E, and so forth and so on. You simply count what you see at that position in that family, and that's put into this system. Okay? That's all. Yeah?
The secondary structure for the entire family is like similar enough, right? So that you can
now, the question here is, okay, if I did that, all right, then essentially it implies, it can only help me if all the 4 other sequences that I put in here have a similar secondary structure. Right? Do they?
or you put different weights on your HDL.
You could put different weights, but still, so if they would be completely different secondary structure, weight will not correct for that. So, do they have the same secondary structure?
Maybe not 100% same, because it's a family, but not exactly the same thing.
Okay, let's. How would you build a family like that? Yeah?
Search the database and look for members that have similarities so that they are well above this curve.
So if we went back to the blue curve, oops, there it was. Oops, oh darn. So we find things by simply staying above that curve, right? We search the database by staying above the curve. If we stay above the curve, we know that any pair in there has a similar 3D structure. And two proteins of similar 3D structure, do they have similar secondary structure? Essentially, yes. This is going back to your question in some sense. But essentially, yes. So maybe there are differences in small detail, but overall the answer is yes, these in that profile. Sorry for the animation, I shouldn't have gone back. So, for all I know, their similarity in secondary structure is beyond the kind of mistake I make from this device. So the assumption that they have a similar secondary structure is appropriate. Again, I choose them such that I believe they have the same 3D structure, and then I believe that essentially they have the same secondary structure. Yes?
So just to complete my understanding, from the fact that they are similar enough, you have only like one hot vector as a target, right? Not like 0.5h, 0.5e, and...
Correct. So essentially the input really is only the count. And you weight them at the same, with the same weight. So they all contribute in this particular case. The question is, are you part of the family, yes or no? If you're part of the family, you contribute equally. Okay? And in fact, every member of the family contributes as much, in principle, as the query sequence. There's a slight caveat with it, that the alignment is built with the first one being sort of a master, and that the alignment is produced such that everything is sort of subdued to correctly aligning the first. But from that moment on, everything you see has equal probability. Okay, meaning the query again has the same as every other family member. Okay? So then essentially we build up the pipeline. In this pipeline we begin with a sequence that we align against the database. We do this pairwise alignment, we use this threshold, we build an alignment, we put the alignment into these two layers of networks. And now we could also put additional information into this. So far, the machine learning device that I presented to you was relatively simple. It used amino acid only. Only just one sliding window. And now it uses, instead of the sliding window, it still uses the same sliding window. Instead of using the simple amino acid that is in one particular protein, it uses the one that is everywhere in this family, what is observed in the family. But what else could we put into this device? So one idea again that I haven't done was the length of the segments. An idea that came up was the composition. So how is the percentage of proline in that protein differing from the average in the database? So this is one thing that you could put in. What else could you put in? What else might matter? So the beauty of machine learning, again, in terms of formulating the probability tables of particular amino acids assigning to particular secondary structures, you compile the probability and that's it. For a machine learning device, you can put anything that you believe helps to solve the problem into the input. So essentially you have features. This is what you have possibly heard of as feature selection. What else could you think about? Yes?
Would you maybe have sometimes information of the kind like where the protein was found in an organism or something like that?
So the question is, do I have information about where the protein was found? So for instance, a nuclear protein. And that unfortunately we don't have at that level. And I haven't tried whether now we have it for more and we can actually predict it at a high level of accuracy, whether that would help. I never tried this. But this is something that I find is interesting enough to try at some point. Unfortunately, we sort of don't do secondary structure prediction anymore. But this is an irrelevant piece of information that we could use. What else? Other ideas?
There were like some properties of amino acids like hydrogen.
So biophysical features. So instead of just looking at the count of which amino acids are in the family, looking at the count of this is an alanine, just look at the hydrophobic residues in a window. Exactly. And that has been used. Any other ideas? Not only hydrophobic residues, also charges and other aspects have been used. Yes? Any other ideas? Have been used successfully. Other ideas? Remember, I told you that proteins have a different scale of length. Some proteins are 40 residues long, and some proteins are 40,000 or 30,000 residues long. And that is nowhere in this input. So So this is something that you may want to put in explicitly. You may also want to put in explicitly when you slide your window through the protein. Are you actually somewhere in the beginning of the protein? Or are you at the end of the protein? Or are you somewhere in the middle? Those types of information are relevant. Then, again, the overall composition is already here. I believe those essentially are the most important pieces of information. And then there are two levels. So the first one does sequence to structure, the second one does the prediction of the first one to the second one. And then you essentially get a bunch of different classifiers or classifications. For instance, one of these solutions that I show here, so each green line is one particular neural network. And the different lines may differ. In one used balanced training, so predicts beta strands better. One used unbalanced training, so has overall a higher level of accuracy. There are different ways. So one uses more hidden units than another one, and one uses other input features. One uses biophysical features, one doesn't. But even a different beginning. So initially you normalize your connections randomly, rather than giving them all the same value. And if you normalize differently, then you would give different solutions. Now, the final solution then is simply an average over all of these individual solutions. It's very easy to show that as long as these gray ones here, they are somehow correlated, that is clear. But as long as somehow the correlation, or they all make some white noise, so they all add their individual white noise, not systematic mistake. As long as they all add an individual white noise, the average over all the grays, the green here, is going to be better than all the grays. Okay? It depends on how much they correlated and how much white noise, so random mistake they make. Again, systematic mistakes you can, by averaging, not get out of. But this is one way. Then today we do much more. Instead of this 2-level, we do a 3-level. The 3-level also uses other pieces of information. I don't want to dwell on this really. This is a device that essentially does the same thing as before. It also predicts solvent accessibility. We will talk about that in 2 weeks from now. And then actually its output is also secondary structure and solvent accessibility. So it just is stacking up the same thing over and over again and sort of cleverly combining the piece of information. Ultimately, this type of device predicts, for instance, it used to be called PhD. So again, the amino acid sequence is running here. That's the observed secondary structure. Blue is the extended state. Here is a short helix in red. The blanks are neither of the two, neither helix nor strand. This is first generation method, Chow-Fassmann, second generation method, Gore, and this third generation method. What you see here, 59 for the first generation is an overstatement, so it does better for this than on average. This does better on that than average. So the question is really, is that this is clearly better here, but is it average? And the answer to that one, or used to be the first solution, that's the distribution here. So this is for all the proteins, the number of proteins shown. So 72 is roughly the average. Now there's something very important in here, and I should have had this idea here. I'm sorry. Let's assume that we had only 2 output units in my neural network. That's dumb, I should have cleaned that up before. So, say I only do H versus other. And say this output unit here is 0.9, and this output unit is 0.1. In that prediction, you would say this is helix. Say this output unit is 0.6, and this is 0.4. You would say this is helix. So the ultimate prediction is the same on this level that I show in the letters. The difference is that in this particular case, the decision is weaker than in this case. Here it is very clear. It can only be the top 0.9, 90% of the probability is here. Well, here is only 60% of the probability sitting here, right? And somehow this information, whether it's a strong or weak prediction, ought to be used. I show this here in the slight gray and the green stars. The green stars are chosen such that whenever the gray number is higher than 6 or higher. No, oh yes, 5. I missed. Thank you. Whenever the gray number is 5 or higher, it's a green star. And what you will see is that wherever you see a green star, the prediction is right. 72 is a number that I look up after I know the observed secondary structure here. The star assignment is completely independent of the structure. I don't need to know it, because this is a number, the starring here, the reliability comes actually from the strength of the prediction. I don't need to know the answer for that, right? So that means I can actually provide an indication where my prediction is more likely to be correct. And this is one of the most important features of machine learning, that you have, however, to work on and to normalize and to establish and calibrate. If we can do this here, then maybe, so for an individual protein, so some proteins are predicted at random levels, right? Some proteins are predicted very high. So if you submit one protein, how do I know whether your protein is going to, I can say on average 72. But how do I know whether it's on that side or that side? Any idea? Yeah?
You could count the stars.
Exactly. Thank you. Now, it's not that simple, but essentially this is the stars, and you see some correlation. So you have more stars, the blue line goes up. And yes, you can use exactly that to see on which side of the distribution you are. Now, on the level of the individual residue, and now the next slide is a little bit complicated. On this level here, I show the percentage of residues predicted, the overall accuracy. Secondary structure prediction is the dashed line here with the circles. And so for the system that I showed, so the overall accuracy, if I predict for every single residue, is 72%. If I only look at the sort of 50% most star-like predictions, so the strongest one, then when I look at the half of the residues that I know are predicted strongest, then for those the prediction accuracy is 86 or 84. So it's substantially higher, right? So this is what I mean by calibrate. So you have to work out, you have to translate what the number, what the star, where do you put the star, what the number that you give to the user, how that translates into expected accuracy. But here is essentially the calibration. And that allows you to completely say which one does work better and which one doesn't. In fact, this device also can do something that is quite remarkable. Here we have a protein that is referred to as chameleon. We have a particular peptide of 11 consecutive residues, and you put that in one position here, it's a helix, and you put the same thing in another position, it's a beta strand. So you have the same 11-mer at two different positions of the protein. One is a helix, and one is a strand. Okay? And this again shows beautifully that the long-range interactions are relevant for secondary structure formation. But in this particular case, and I'm not going to go through this slide again, but the prediction method that I show could actually predict this. It saw that the prediction in one position, here was a helix, and another position here was a beta strand. Prediction was right. So using this evolutionary information could even help. And what I have to say here, the window that the system used was initially 15 residues. So this is an 11-mer, which means it covers almost the entire window. It does not see much more. And it can correctly predict it simply by the evolutionary signal. So that gets us into a new theme somehow. This is not about machine learning, but it's about how to assess methods, assess publications. Say you read a paper, and the paper said, I have a new method here, and the new method called B has a performance of 63%, while the best method that existed before was method A, and that is 60%. I am much better. You're the reviewer. Is that statement true? So, just to make sure, I understand that 60 minus 63 gives 3, and that 3 is larger than 0. This is not the level of our discussion. There are a couple of questions you have to ask as a reviewer. Can you shoot? Yeah?
Well, the other things being equal principles should be.
Say again?
The principle of other things being equal. So everything else, the setup of experiment, the data, everything else.
So this is and this is in fact longer than you may believe it is. So he says that everything has to be equal, the setup of the experiment. So funny enough, you'd be surprised how often publications, they did not talk about the same way to compute the performance. So one says, well, I got 63 in computing how good I do helices, and the other one got 60 in the Q3, so overall accuracy. That may sound funny to you, but I bet if you have a long career as a reviewer, at some point in your career you will get across a case where you will miss that sort of thing. Because people by mistake or by design try to fool you into believing that this, of course, cannot be the case. The next issue clearly will be the same data set. Now, the same data set, what does it mean? Let's look at this example here. Both these methods used 100 proteins. Both used random splits, and both took half for testing. Is that as much as you need to know about the data set? No. Exactly. Exactly. So this is absolutely not enough because you don't know. This says random, so is it the same 50 or not? You don't know, right? The other thing is, why those 50%? Why not the whole thing? Ultimately, they have to do, if that's one cross-validation, they have to do the other round. They have to do it again. So when they do it, you need results that in fact say every single protein in my data set has been used exactly once. Not randomly, some of them twice, some of them once, everyone exactly once, okay? So that we know that the 63 is defined in the same way on the exact same data set. Now, there still is an issue. The next question is, even if they gave you the results for the same 100 proteins used for testing, how exactly did they split? And what are these 100 proteins? Are these 100 proteins such that there is comparative modeling possible for any pair? So say in my set of 100, for the sake of the argument, say 66 of those would be sequence unique, meaning 34 would not be. Maybe in one of the two, 17 were always in one set of the training and 17 of these in the other set. So essentially it learned comparative modeling, right? And those could be, since you already know that some proteins are predicted better, those could be the ones that completely determine the +3 percentage points, because they could have moved the structure. So we have to see that in fact both used a sequence unique subset. The 100 are really, there's no overlap, and the way of splitting cannot be the issue. But now, say this creates the same conditions. Say this creates what you said. They use the same conditions. And again, the question is 3 percentage points better? And again, so in most publications, this, in our field and other fields, this is not what you will have in front of you. So this, what I describe here as trivially has to be the case, usually is not correctly done. But now comes the next issue. Even if it is 3 percentage points a number high enough to say it's better? Yes?
I think not, because the confidence is
So this is, we will get back to that. It's a very, very important question here. So how does that compare to random? So I have a 3 percentage point increase of a number that is 60%. If random would be 50, is that really a significant improvement? And that brings us, in a more generic way, to the question, is it significant? Now, whether it's significant or not somehow depends on the details. So one way in which you can look at that is as a distribution, right? So here we have, I showed you this distribution with 72 as an average. There is this distribution. In this particular distribution, can you visually sort of guess what a standard error would be, or standard deviation, or can you sort of somehow, when you saw some figure like that, and the author would not provide standard error and standard variance deviation, do you visually know what a standard deviation is?
Here you're saying it's 10.
Oh, that does help if you read the slide. Yes, I would of course require that I remember what I put on the slide, which in this particular case I didn't. So visually, so I say 10 up there. So visually, you just have to find a point where you believe about 66% of the data sit, right? This is somewhere here. And the plus minus is the standard deviation for where 66% of the data sits. In this particular case, about 10. How does that translate to standard error? So the sort of rule of thumb of that is you take the standard error, the standard deviation, you divide by the square root of the number of points here, the number of proteins, and you get the standard error that in this particular case is plus minus 1. So this plus minus 1 then would give you 90% of the distribution. So if you're plus minus 1 away from the average, then for 90% of the data you can say that is significant. If you stayed away 2 standard deviations, 99%, so forth, right? But within the sort of 1 standard deviation, 3 percentage points is higher than 1, right? So it's at least 3 percentage points is above the standard error. It may still not be in terms of background of random. We get back to that, but at least for the standard error, in that sense, you would say, okay, that statement checks out with the standard error. Yes?
Could you explain why it is 19 or 66 again, when the standard error of 1 sigma left and right?
Sorry, what is my mistake I made? What is?
I mean, you said 90% of data is covered by 1 standard error.
No, plus minus, it's 2.
2 is 90%?
Okay, here's another question. So, in this particular case, I should have put it the other way around. Say method B would be 20 years. So the difference between method A and B would be such that method B is 20 years older than method A. Could B still be better? So B again was the one with 63, A was with 60, but B is old. Can I hear something from the reviewer? Yeah? It could be simpler, yes. Simpler good?
Sometimes no, but our understanding of how proteins work just changed over years, and it's not standing at the point right now. So
No, okay, yeah. You have another point?
Well, it's basically like he said, if something is proved in the post 20 years, we cannot exactly compare them. B is going to be invalid.
So the statement here is B is going to be invalid because it's much older, and since 2 methods are completely from different time periods, we cannot compare them. That is right in many ways. So the method A has had the chance to use many things that method B has not used. But I would completely debate the statement that we cannot compare them. It's rather the opposite. We can. And actually, not only can we, the number is what should count. How old they are doesn't matter. If anything, you may say, well, because method B is older, it has used fewer things, and because obviously it had to be simpler, because then fewer things were there, and it still does do better. but actually may give an additional an advantage. But that's all nonsense. The only thing that you have in front of you are the numbers, and the numbers have nothing to do with the age of the method. And the numbers in this hypothetical example, typically it's the other way around. And I have to change my example here. But typically, what really only counts is the 3 significant, yes or no. And one way of saying significant that we discussed so far is standard error. By the standard error is significant, whether it's 20 years old or not, at least by the standard error is important. So if it's statistically significant, the age doesn't matter. Is there any other test that you can do? Mind you, B is 20 years older. So one thing that you can do, I'm going to give a different solution on the next slide, but one thing you can do is you can see whether method A performs differently on the things that have been added over the last 20 years, right? So over the newer things. So meaning that method A, although it's less accurate, but maybe even on the newer things is more over-optimized or not. But ultimately, age apart, you can try to see, so you developed your method, we began developing your method, so you extracted the data set of 10,000, that is today. And then typically you do the machine learning and do all kinds of things, and then the thesis at an end, and typically this is half a year later. And at that point of half a year later, the database has grown. So you could find the ones that have not been in the database when you started all of that, and simply assess, you have 2 methods, assess both methods on these new proteins that none of them has ever seen, provided they are not within a comparative modeling range. So they're distant enough in terms of sequence, then you have somehow a new data set. And that gets me into the cross-validation how to do, and are there any questions in the room? So I have 2 more minutes. 2 more slides. This is an important thing. I will bring it up again. Let's begin with it today. Okay, so I have one way. This is the entire data. Say the entire data had 300 proteins. I take 150 for training in one cross-validation and 150 for testing, and then I turn it around. So I use everything. And then at some point, I find that for 15 hidden units, I get a Q2 of 62, for 30, 64, for 45, 63. And again, I do the same thing in the other direction, right? So I take this half for training and this half for testing, and those are the averages over 2. And then my conclusion is the best performance is Q3 is 64%. Is that right? I'm currently not open for input because I'm seeing that my next slide simply asks the question, is that right? So?
Yeah, I think it's not because you're fitting your result to some data.
Yes, that's the crucial point here. So test set means you make no decision on that data set. The only thing you use the test set for is to assess the number that you will publish. What we do implicitly by looking at the table is we make a decision. We have 3 choices of hidden units, and we ask which of these 3 is best. That means we use the test set that we are supposed to have under the table, we take it off from the table, look at it, and use it to make a decision. Yes, the best method has 30 hidden units, and then the next statement, and the accuracy is 64%, is wrong. Because this number, 64, is only right if you would not use it for any optimization. How can you get out of that? Well, one way of getting out of it, you have a sort of 3-fold split. And again, in the literature there are many names. I use this word of cross-training simply because from the way it sounds, you immediately realize this has to do with training and development, right? So you take, for instance, the way you split in detail here, whether it's 100, 100, 100 or some other way, doesn't really matter. And there's a lot of additional things to be said about that. Let's assume we have an equal split. So this one here, you use to optimize the connections. This one is in fact exactly the one you choose for picking. So you say 30 is best because for the green set is 64 and 30 is best. You may be inclined to actually, you're not supposed to do that, look up how do these numbers actually look for the test set. And you may actually see that what would have been best for the test set is actually 45, not 30. But then what you have to publish is my best solution has 30 hidden units and the performance is 61%. Although you somehow seem to have in your data evidence that you can do better, but this again would be over-optimizing or would be essentially over-tweeting your parameters. As long as whenever, you should actually never see that, but if you really saw it and then write 30 and prediction accuracy 61, you do the right thing.